Okay, the next presenter is Rzepiela from Pyxis Discovery, which is a Polish company. Anna studied initially in computational chemistry and latterly in computational physics in the Netherlands. She's now working for Pyxis, and she's here to give us a talk again about compound suppliers.
So first of all, thank you for the organizers for inviting us to present here. I'm gonna present a platform that is developed by Pyxis Discovery. It's a commercial platform for compound sourcing. Just a few words about Pyxis and correction to the introduction. Pyxis is actually a Dutch company. We are located in Delft in the Netherlands. We are a 5-person company. Our specialty is computational chemistry-based services. The other area of our interest and activities is design of small targeted libraries. So those are compounds for biological screening that are available on shelf. And recently, new activity is the platform I'm going to talk about today. So what was the goal for Chemonaut? This is historically a database that we have been maintaining in-house for our services, for projects of selecting compounds for certain screening campaigns. And recently, about not yet a year ago, we have launched it as a web-based service. The idea is to offer compounds from reliable suppliers. So the standard we have there is companies have to be able to deliver the compounds within three weeks. They have to be able to supply QC data for their compounds, and they have to have at least 70% of the structures in their database has to be unique. So we don't want to deal with compound collectors that buy their compounds from yet another source. So what I'm going to talk about in a little bit more detail is how we standardize data that goes into the database. Then also I'm going to talk about the filtering we apply. So the major use for the database is to select compounds for biological screening, and therefore we filter all the compounds based on lead-like rules that we have developed in-house. And I'm going to talk about this also in detail. And all the structures in the database have pre-calculated physical-chemical properties, so those can also be used for filtering or sorting of the database. So very quickly, general setup. Chemonaut is a database, an Oracle database that communicates through ChemAxon JChem cartridge with either Eastern JChem, so local application, or through in-house written scripts with web clients, so you can access the database through a website. And just a few screenshots of how the website looks. So this is not working. So anybody here knows this drawing applet? You can draw a structure. All the typical ChemAxon structure possibilities are available here. And then we have a number of databases that you can select from. Let's say default selection is screening and building blocks. Then we also have special libraries. Those are libraries from suppliers that are generally more difficult to get access to. You have to have a good relationship with the supplier to actually get those databases. And there is also a virtual library. So those are compounds that suppliers think they can make relatively easily. So as the first screen, you go to the search window, so the search tab. There is also results tab, which I will show later. You can save your queries or selections, and you can send inquiries. In the options for searches, so you can filter on number of pre-calculated physical-chemical properties. And you also have access to the, let's say, advanced structure searches supported by Marvin. And this is a typical result page. And a few things to notice here is that as soon as a compound can be sourced from multiple suppliers, in the supplier field there is a drop-down menu that you can select from which supplier you would prefer to get this compound. If the salt data for that structure differs for any of those suppliers, this will be adjusted automatically. Then you will also see the corresponding ID number. And if available, also the availability data. If any compound is available from just one supplier, then there is no drop-down menu, and there is just that one name. So, in general, the procedure for importing the catalogs from suppliers into the Chemonaut database consists of two distinct parts. Is how we treat the structure, through what kind of workflow goes the structure, and then the data. We limit ourselves at this point to storing the supplier ID, salt data, supplier name, library name and type, and whenever possible, and that not always comes in an SD file, sometimes in Excel, sometimes in text file, availability and pricing. So what I will focus in detail on is this part of the workflow, the different standardization steps that any given structure that goes into Chemonaut is going through. So for this we are using the ChemAxon tools from Standardizer, but also other ChemAxon tools. So things we have seen before today, ungroup abbreviations, remove explicit hydrogens. We disregard any structures containing query elements or macros elements. We want a real structure with a real ID, so this is not what we want to store. Then we've seen some of those today as well. We have seen nitro groups as drawn in many different ways. We want to have it stored in a database just in one standard form. Another example is the sulfonyl group. Again, transformations that are being done on the fly when the structure is considered for import into Chemonaut. Another thing that we also look at is the charges. So if in the structure field there are two structural elements, and for example there is a charged nitrogen and a charged oxygen on a bigger organic compound, then we neutralize both the nitrogen and the oxygen. So we prefer to have as the standard the neutral forms of the compounds. Again, if there is a charge on a salt that is in the structure field, but there is no corresponding charge on the molecule, then we fix it in such a way that the total charge in the structure field is 0. And a similar example here, if there is a charge on the molecule but no charge on the salt, then this is also being fixed. So let's say the standard way the structures go into the database are also defined. Then we look at the salt in the structure field. The way we do it is, again, look out from a salt table, so not using the standard options from standardizer, not looking for the biggest or the smallest or the biggest, the larger in terms of atom count. So there are a number of ways of looking for the salt in the structure field if there are two elements. We do as table lookup. Things that we disregard if there are three elements in the structure field, they are all three different from each other. So it's, if there's two different salts in the structure field, that's being disregarded. Again, example that we've seen before. So then after the salt, after all those manipulations of the steps of standardization, we look for valency errors, and some of them can be fixed. And if so, we do so. But for some less commonly seen errors, we disregard the structures. So they are not imported into the Chemonaut. What we do, we write out an ASIFA of structures washed out, so we can review them and improve the workflow and include those cases as well. The thing that so after structures has gone through the standardization steps, what we do to this screening database, so not the building block database, we also filter for structures that have been reported in literature to frequently show activity in biological screens, but have turned out to be false positives because they are acting as covalent bondings, electrophiles, suicide inhibitors. I list here just a subset of the literature that is available on the topic. The total number of filters that we have is right now something over 80. Some examples here. This is all stored as smarts. So this is also all done on the fly, on the import of the structures into the database. And again, there are a number of classes that you can divide those filters into. So chemically reactive, biologically unstable, or in general reported as difficult to optimize in the drug discovery process. So this is where we are now. This is how the Chemonaut database is constructed at the moment, and this is what you can search via the website. The plans that we have for the immediate future is to include Protomer searches, and we had two very good talks yesterday about that. So we're going to consider all the options that you have there, whether to enumerate or whether to do it on the fly, what's the best way to tackle this. The thing I'm going to touch upon just now is generation of ionized macrospecies for the structures in the database. And the thing that we would like to do in the future is also allowing users to either download the search results as 3D structures, but also maybe in the more further future also allow 3D searches. So what I'm evaluating right now is can we use ChemAxon's pKa prediction as a tool for enumeration of ionized macrospecies of the structures in Chemonaut database. And the idea is if you can do so, you can perform more advanced substructure searches. You can, for example, look for structures that contain both a basic and acidic group. So like here, this deprotonated group and a protonated group in the same molecule. There are a number of other tools that can do it right now, it's like blick prep or wash in Mo that will deprotonate and protonate groups. But they are in general limited into what they consider as the groups to operate on. So the primary goal for this is to allow users to really do more advanced searches, more advanced substructure and full structure searches. If you know, for your screening, if you need to have a protonated group, this is the way to go about it. I'm sure that most of you have seen this interface of the pKa predictor from ChemAxon, so you can run a prediction from, for example, Marvin view. Then you get a pop-up window with the options for the calculation. Here is just all the defaults, and then you get a window for what is the acidic pKa and the basic pKa, and here you have all the protonated and deprotonated forms of this molecule that can exist, and a very nice chart of what are the distributions. This is per molecule approach, so it's very nice, but you can look at one molecule at a time. So you can actually run the same prediction, so just to get the values of the pKa, so you can run it through the batch mode, so the CxCalc, and you can define, again, this is the default, and the minimum pKa that you want to get as a result, the maximum pKa, how many of them you want to get, and your input and output files. So this will calculate just the values. So this will not generate the protonated forms. In general, in literature and also in the poster in the lobby, the consensus is that Marvin performs quite well as compared. This is a recent paper from AstraZeneca. At the time I was looking at it, it didn't have page numbers yet. So in general, the RMZ is low for Marvin, and it also has very good detection score in terms of how many of the protonated and deprotonated centers it can predict. For my use, so for our use, what's important is if we generate the macrospecies based on the pKa predictor, what's the increase in database size? How many more structures do we have to store to have this information available for searching or downloading? And if you look at the numbers, so again from this paper, the number of basic groups that were measured is 135, acidic 7. If you run the pKa from ChemAxon, you get quite a lot more with the standard settings. Or if you use different settings that might be, let's say, experimentally and biologically maybe more relevant. So most of the experimental methods will deliver results in pKa range between -2 and 12. But this is also generally the range that is most interesting for the biological systems. But still, the number of predictions, it's staggering comparing to what has been measured. So there are too many of them, if you ask me, for my purpose. So, but like I said, with the pKa prediction, you get just the numbers, not the protonated forms. What you can use for generation of protonated form is this function, again from the batch mode. And you have to specify the pH at which you want to see the macrospecies, and again, input and output file. So in general, my experience with this is that actually using this function, you get even more protonated and deprotonated forms than what you got in the table with the values. So there's even more of those forms. There is no handle on the pKa range. There's no handle on the pKa numbers, so how many acidic and basic groups would you like to get out of your calculation. And a lot of the protomers that are being generated have the percent distribution virtually 0. So they are basically unimportant from the practical point of view. So I've been discussing this with some of the ChemAxon guys, and they're going to add some of those features to be able to limit the number of protomers generated. So just to close, Chemonaut statistics at the moment, we have in unique structures, we have 2.5 million unique structures in the screening database. We have almost 500,000 unique structures in building blocks and special database. So those are the ones that are a little bit harder to get a hold of. Again, almost 600,000. Overall, it's a bit more than 3 million from 51 suppliers. From our estimations, this actually covers quite substantially all the possible chemistry that you could be importing into your database. But keeping in mind again that we want to focus on the chemistry that it can really get, that there is QC on it, and that the delivery time is not outrageous. And next to that we have virtual library with 11 million structures from 9 suppliers. Those are structures that should be possible to synthesize in not too long time. And this is where I end, and thank you for your attention.